Gian session. So it's the last day, and here we are having only lecture in the morning session and two lectures in the evening session. You can have interaction after the lecture. First lecture, you will have a continue probably one lecture and then you can have a break and then interaction or lecture depending on this slide. So you can interact. In the afternoon also we have seen like 3 to 4 lecture and interaction that we can do. Just after the lecture today at 4 we will have some interaction feedback with the foreign faculty that you don't miss the protocol of how we call that we can do after the lecture. When all the lecture is completed, the students are participants are required to have some interaction with the foreign faculty in terms of feedback. We don't have proper time like after the 4 you will have some feedback, then you will 4:30 or 5. So there is an exam for 1 hour tomorrow. So you have to, I think we will especially arrange it for tomorrow 1 hour 10 to 11, sorry 10 to 10, 10 to 11. If you are comfortable, can we do 9 if you like in the morning so that you will be free later. But 9 will be too early for you and this thing also. So you will take comfortably breakfast and come at 10. Right? So 10 will be fine.
Yes.
So be careful that you all should attend the examination that is 1 hour and should be like your certificate will be evaluated only if you are attending this. Alright?
Yes.
Any issue anyone with this? Nothing? Yeah. So I welcome Professor Todoro to give this talk on the last day and take the maximum benefit interaction out of the lecture. Thank you.
Good morning, everyone. How are you feeling like last day? Already tired? Too much information? Well, don't worry. Soon we'll be finished, but the bad news about the exam. So, even the good things, they have the bad things after that. But again, it's maybe it's someone will come in my lab after that to spend some time there to some exchange. Yesterday we had a very nice conversation with Vice Chancellor of the university. So he was giving a good indication that he's interested in establish the working collaboration between our faculty of Pharmaceutical Sciences of Sao Paulo University and your university. And most probably even after that we'll go for further for agreement between the two universities. But first we'll explore our faculty and your university because it's something that it's allowed for the international relation. And well, let's continue. So today we're going to move a little bit more on the probiotics, not such topics too much bacteriocins, but again, I believe that bacteriocin production actually could be a good characteristic of the probiotic strains. To be a probiotic, don't need to be a bacteriocin, but if the probiotic has bacteriocinic potential, bacteriocin production, this is a good addition to the activity of the probiotics. According to the definition that is accepted, was suggested actually the World Health Organization, and probiotics are supposed to be alive cells. This is very important because very often people say, yeah, but they can be not really live. From then even some people trying to put even prebiotics, that is a chemical structure that are used by the probiotics as a probiotic. So the definition is very clear. Need to be a live microorganism, then giving the benefits for the cost. And the probiotics, in fact, could be, and they need to be in an adequate numbers. Now, what number need to be? different agencies, different countries, they adapted the different rules. Some say 10 to 8, some say 10 to 9, some countries say 10 to 7. Some even say if they're enough and they do their job, no limit. I mean, could be any numbers if they do their job. Because obviously, live cells, they need to be in some quantity. uh, Beneficial microbes, they're not like the pathogens. You know, when you look salmonella, shigella, escherichia coli, sometimes they're enough for really a few cells to do their job. I mean, even 10 cells, they're enough to make you sick. Probiotics, they need to be a little bit higher numbers, and they need to be in adequate numbers in order to perform their work. Because remember, when they go via our gastrointestinal tract, they will compete with others. And some of them will be not able actually to reach the place they need to perform their beneficial properties. So we can have the probiotic cultures from two different ways. You can have them from the different supplements from the pharmacy, that, or we can have them as a part of the microbiota of different fermented food products. So technically, I'm much more in favor of the fermented food products as a delivery, but then it's coming up a little bit technical problem. How are we going to be sure that the bacteria that is a probiotic bacteria, it's enough in these products? It's much more challenging because you have the natural microbiota, you have the condition of storage, all these things, you have the shelf life, and you need to be sure that your beneficial microorganisms is inside and it's enough quantity and right bacterial culture. So in the last year, in fact, probiotics actually going much more to the pharmaceutical products because very often in the food products we cannot guarantee enough number and up to the end of the shelf life. But again, still it's much more pleasant to have the yogurt or cheese instead to take the pills, right? But again, just remember that the probiotics are not medicine, they're supplements. They are there to help to improve, they help to facilitate treatment, but they are not medication, they are not pharmaceutical product that will cure your disease. So some supplements, they can be applied as these products. And we're also already discussing in several occasions in the individual conversation. In fact, this kind of packaging for the probiotic bacteria, I'm a little bit skeptic that they are proper. Why? When the probiotics are lyophilized, they are very sensitive to the oxygen. Any bacterial culture lyophilized, oxygen is the biggest enemy. So imagine you have this packaging and you open the lid and you have the film that is protecting. You're removing the film, but then it's over. Oxygen can go in, and you have it, let's say, I'm not sure this exactly, but let's say 30 or 60 capsules. And you're going to take 1 or 2 capsules per day. So for 1 month, this will be exposed to the oxygen. So I'm much more in the favor of the other type of packaging that every capsule individually packed, because according to the pharmaceutical industry, it's supposed to be under the modified atmosphere and it's much more protected. Because it's very important to have this appropriate number till end of the shelf life. Another important thing in these probiotics preparations, they need to be very often keeping the cold temperature. It's well, on the package supposed to be written, keeping cold. In the pharmacy, normally they're very strict in their doing, but very often people taking the cold, they don't care, they put in the normal temperature, and this could be, again, some problem. So, let's go further. Okay. Most of the probiotics that are in the market, they belong to the group of the lactic acid bacteria or bifidobacteria. Even some examples of Saccharomyces. And in the last couple of years, we have this as whole new generation probiotics. So bacterial cultures that are normally no one was looking before on them, they appear to be very important. Akkermansia, it's a very interesting strain that is strictly anaerobic and is part of our gastrointestinal culture. And the modification of Akkermansia or some other, even some Clostridium, in fact, can interfere with our health. So we need to keep them balanced in our gut. So in some in several situations, we need to take care to having these strains, and especially when you develop some diseases, restoring to gastrointestinal balance is important. It's not just the lactic acid bacteria there. They're good players, they're showing their beneficial properties, but they're not predominant microbiota in our gut. We have several other groups. But discovering the problem, because some of these as called new generation probiotics, several of them is very difficult to reculture in the laboratory conditions. So, well, biotechnology is supposed to find a way to grow them. Then maintenance, strictly anaerobe. Obviously, this kind of packages for strict anaerobes will be not an option. Come on, move it. Okay. Why they were good? Lactic acid bacteria, there was first one explored because of the historical point of view. There was much more easy to culture. There was already known by the fermented food product that they have some benefits. So they can produce the different acids, they can produce diacetyl, hydrogen peroxide, of course, bacteriocins. They can improve the organoleptic property of the fermented food products, so making it much more easily and convenient for early exploring. At the beginning, even it was even more strict because I was telling that the probiotic bacteria needs to be strictly isolated from the human or from the other animals and apply as a probiotic. But then start to publish the results and showing that fermented food products isolates lactic acid bacteria or bifid or Saccharomyces, etc. They can be a good candidate. So these things, this story that some reviewers always complaining, but you're reporting that probiotic isolated from fermented food product is not human origin, so cannot be probiotic. It's not true. Any bacteria that are not toxic, that bring the beneficial properties, and they is able to survive in gastrointestinal tract and safe, providing something good, why not? We can explore, we can make arguments and prove that this culture could be beneficial. No? Okay. Even I think even the pointer is getting tired of 5 days. Normally, the problem with the probiotic is coming with the fact that they are good. We know that they having beneficial properties. And very often they are recommended to you by the medical doctors or by nutritionists. But the problem is coming with the fact that very often people self-prescribe the probiotics for themselves. I mean, you talk with a neighbor was telling you I had improvement of constipation because of taking this probiotic. You can take it, you can be good as well. Yes, this is not a bad idea, but we need to take attention that the probiotics are very specific. And they can have the beneficial properties for a specific group, but can be maybe problematic for another group. So always the opinion of medical doctor or pharmacist or nutritionist, it's very important to be taking the probiotics. As well, maybe you're taking some other medication and your neighbor don't take this medication. So obviously this can interfere as well. Because it's observed in several situations that some probiotics can have some side effects. Let's think about the following scenario. You have the Leuconostoc mesenteroides as a probiotic. Leuconostoc producing exopolysaccharides, right? But the exopolysaccharide, they are carbohydrates. And if you have diabetes, maybe this kind of probiotic not going to be good for you. There was some cases described in the literature showing the woman taking the probiotic that producing exopolysaccharides. In fact, she was having high level of the blood sugar all the time. And this is an interesting point to showing that some probiotics are not really appropriate for some group of consumers. There was some indication that the lactobacillus now is, I think it's, sorry, I'm not sure what the new name of lactobacillus rhamnosus. No. Okay. This species is not recommended for pregnant women. And it's not recommended for people with a bowel disease. But very often, it's not written on the package. Medical doctors are supposed to know this and supposed to tell you if you are having a bowel disease or you're pregnant, please don't take this probiotic. But well, why not? This company is not putting this attention, because very often from companies producing pharmaceutical companies especially, they don't want to make you scared. They put you only the good part. But again, it's a responsibility of the industry to put even some potential negative effects. Very often, this is written inside this huge paper that's coming inside the box. How many of you read this paper, huge paper, when you buy the medicine? Very often, no. People normally start to read the first few lines and then quickly go to the text, look for some specific area, and then stop it. But it's important because very often some additional information is there that need to be taken in consideration. Um, we already know probiotics can have the benefits. They work. But again, need to be a right strain or a right person. And they can strengthen your immune system, they can be combat against allergies. There was a probiotics developed for the people taking chemotherapy because it's improving the vitamin production. There are some probiotics producing vitamins from group B. And when you take them, you're improving your gut balance. Imagine you're doing chemotherapy. How many substances you're taking? You're taking radiation. You're taking. It's a very complex process, and this destroys your gut microbiota. And you need the probiotics. Well, probiotic in this case is not care. It's not care for the cancer, but it's care for your intestinal balance. And in this way, they improving your quality of life. And it's helping. If you're healthy, if you have a good physiological status, your chance to fight against cancer is better. So in this way, they was working in synergistic with the other methods and improving your chance to win this war against the cancer. So they can improve the reduction of some pathogens. Just to simply think about it, if you have a metro full of people, it's coming a bad guy. This bad guy have the chance to enter in the metro? No. But if the wagon is empty, the bad guys coming and they enter and do what they want. So it's the same thing, a very literature example. But if you have a good probiotic microbiota, your natural intestinal microbiota is in the good condition, and you got the environment is full of these beneficial microorganisms, when it's coming the pathogen, he not have chance to compete for the receptor of the mucus and to perform. And even antimicrobial compounds produced by the probiotics will be able to reduce chance of the pathogenic bacteria to survive and to perform negative effects on your body. Story, in fact, starting with Ilya Mechnikov and the definition for life probiotics. It's actually related a little bit later, as a term, but Mechnikov and his collaborator, let me see, I think I put in the next slide. Yes. This actually is a Bulgarian medical doctor, Stalin Grigorov. He was working together with Mechnikov. Very interestingly, Mechnikov never been to Bulgaria, and he worked with the Bulgarian yogurt. And he even claimed that the Bulgarian people eat the yogurt and they stay healthy for a long time. How he know this? He's never been to this country. And that time was no internet, beginning of 19th century. So Stalin Grigorov, in fact, was working in Geneva, and he met Menshikov in a conference at that time, and he brought some already isolated lactobacillus bulgaricus from yogurt, and offered to Menshikov, let's study this strain, and they start work on this strain, showing their beneficial properties at that time, in a, obviously in very primitive way. I mean, you don't have PCR, you don't have NMR, you don't have sequencing at that time, but they was enthusiastic in showing some beneficial properties. So, interestingly, after that, Menshikov took all the credits from his collaborator, and all the world now know Menshikov, but don't know a real person that go behind this. Well, it takes a little bit more, let's say, political story. Bulgaria and Soviet Union, they was in the same bloc in the past. So obviously Bulgaria don't wanted to break the word of Big Brother, the Soviet Union. So at that time they was agree, okay, let's be Menshikov, let's be Menshikov, let's be Menshikov. But again, now in these days we actually looking for historical and scientific truth, and actually wanted to promote his work. And he was actually working not only on the Lactobacillus bulgaricus, but again using penicillium modes for the treatment of tuberculosis. At that time, this was one of the options. Later, between the two wars, he received an offer to be the director of Pasteur Institute in Brazil, but he refused and decided to continue working as a medical doctor and help to the patients. Well, Menshikov, it's a Russian, Ukrainian, it's a very big discussion because he was working in Kharkiv University, that indeed is in Ukraine, before it was Russian Empire, after it was Soviet Union. Well, but it's some confusions of the geographical names. Little bit later, Henry Tisser was isolated bifidobacterium, showing that in fact bifidobacterium can be a good probiotic and can bring some beneficial properties. Even some of the early applications of the bifidobacterium was showing that they can really strengthen the immune system of the patients. Now it's coming to a small confusion with the bifidobacterium. Well, bifidobacterium producing is a very good acid production. And in fact, this can make some problem for dental problems. Because if you have bifidobacteria too much in your mouth, it's not a really good idea. But in fact, which time of the human development actually bifidobacteria is more important for the babies? Because bifidobacteria playing a very important role in the utilization of exopolysaccharides that coming from the mother milk. And the baby have the dental devices in the mouth. No, they come a little bit later. So you see, the nature was structuring very well how can work and when can work. So the baby don't have teeth, and having a milk from the mother need to utilize the exopolysaccharides from the mother and having bifidobacterium in the mouth and in the gut in order to produce these enzymes to break down oligosaccharides, and to have them as a single sugars and to use it. These days, so when we apply the bifidobacterium, we need to pay attention because they're not supposed to very long time in the mouth. They're supposed to go and colonize the gut. So in some cases, capsules are more appropriate or in, let's say, encapsulated bifidobacteria because they need to pass the mouth and go to the gut and do their job there. So sometimes these kind of dairy products enriched with the bifidobacterium, maybe it's not the best option. Or maybe needs to be, you see, some questions about the probiotic, they was developed, they was suggested, but then appearing some problems, they need to be solved. So as a food technology people, we need to think sometimes how we can avoid these potential problems and put the emphasis on beneficial properties. Because in the nature, nothing is just a black or white. Everything is between and depends on the consequence and the situation. Okay, now it's coming next step. Alfred Nisler in 1970, he isolated Escherichia coli. And this strain is one of the very iconic examples how a strain that is considered in general as a pathogen, strain that is considered a big problem, Escherichia coli, could be even beneficial. And this strain is more than 100 years as a production as a probiotic. The story is very interesting because this is the First World War. Alfred Nisler observed that some of the soldiers cannot develop shigellosis. And he donated from the soldiers that are not struggling with shigellosis. Well, at that time, Shigella was not yet described as a Shigella, but today we know that it was Shigella. Is showing that, in fact, they had this strain of Escherichia coli. And this strain was actually helping to prevent of diarrhea and development of infection with some other gastrointestinal pathogens. This is a very important thing to mention that not all Escherichia coli are bad guys. Some of them are full of virulence factors, some of them are extremely highly pathogenic, some of them can kill you because of the intoxication, some of them producing toxins, some aggressive hemolytic, etc. But some strains, they actually could be beneficial. We're having a high number of Escherichia coli in our gut, and they say our Escherichia coli, and they're responsible for production of vitamin from group B. I think it was B12. Oh, now I'm confused a little bit. But about vitamin B group, they're producing there because our body cannot produce these vitamins, and we needed Escherichia coli, our gastrointestinal strains, to produce these vitamins. Well, and this strain particularly was showing very strong beneficial properties. We was doing a kind of survey of salmonella in Brazil, checking the in meat production several more than 4 years. Actually, 4 years we was checking more than 20,000 isolates of different salmonella, and more than half of them, there was not all virulence factors, nothing. There was completely not dangerous. Some of them was very dangerous, but some of them just nothing. So again, species could be considered as a safe, considered as a problematic, but we need to go to every strain-specific conditions in order to explore and to check these problems. In 1920, Röntgen demonstrated actually that this matching of Bulgarian bacillus, in fact, don't colonize the gastrointestinal part. And they come in and they go out. At that time, a little bit after, was the principal idea is that probiotics supposed to colonize and stay there to do their job. Today we know that it's not really needed. Some beneficial microorganisms, probiotics, they can be entering, they can do very quickly their job, and they can go out. So it's everything depends on what you want and how do you want and what you target. As I mentioned yesterday, if you target immune response, obviously you want a colonization. But if you target just the removing of heavy metals or mycotoxin or some other toxin, obviously quick passage of this microorganism will be good. Um, Everything started to be more and more focused on this area. In 1935, certain strains of Lactobacillus acidophilus being isolated and showing that they can have, because interest about these beneficial food products and beneficial microorganisms start to grow. And in fact, term of probiotics was quoted in 1953. Constructing antibiotics, probiotics were defined as a microbially derived factors. And in 1989, Roy Fuller suggested this definition that is more or less accepted today. A live microbial feed supplements which beneficially affects the host animal by improving the intestinal microbial balance. So need to be live, need to be with the feed, or okay, for the animals, food for the humans, and needs to have the beneficial, we need to improve intestinal balance. Well, in this case, what about probiotics for the skin? They are not food. So you see that the definition actually slightly needs to move, and it's already moving because these days we have a different application of the probiotics. Okay, with some of them I agree, some of them I disagree, because if you can prove that, well, we have several microorganisms on our skin, this is completely normal. And if we can have this microorganism additional on our skin and they can provide the beneficial properties, perfect, this is a good idea. However, trying to convince me that you can have a salt with the probiotic properties, I really disagree with this point. Because the salt have a very long shelf life, it's a very aggressive condition, how the microorganism will survive there. Well, it's difficult. If you have the good argument, convince me. I'm open for this discussion. But I really was very skeptic that microorganism, probiotic, will survive in the salt. In my opinion, they cannot survive in shampoo. They cannot survive in lipstick. They cannot survive in the beauty creams. Maybe encapsulated, but it's not for this extended shelf life. It's kind of strange. But then it's coming the point. Maybe we needed to change because we know that some of the dead probiotics, dead cells can have beneficial properties. So this later appeared term postbiotic, where it's the dead microorganism, but they still have their beneficial properties. We're going to have some more about this topic. In 1960, dairy industry adopted the idea and they discovered the magic word probiotics. It's selling everything. You just put probiotics on the label, we'll sell you product as festive season candy. Everyone wanted to buy. But again, several of other strains being at that time incorporated Lactobacillus rhamnosus, Cassie, Johnson, etc. And all these products become more and more popular. Industry not always respect scientific recommendations. Very frequently, these products are not containing enough microorganisms. This is a problem because they cannot have appropriate beneficial properties. So this is a story that I was actually telling. You have a school bus full of people. Beneficial microbes already installed in your gut. It's very difficult the pathogens to come and perform. And if you have empty bus, it's very easy for the pathogens. It's a very simple example how this can work. So what could be a beneficial properties of probiotics? We could have managing lactose intolerance, prevention of the colon cancer, etc. Well, lactose intolerance is a very important point because normally Indo-European race don't have a problem with lactose intolerance. But if you look for the Asians like Chinese, Japanese, Korean, you look for the Africans, you look for the Native Americans, they're having a strong problem with the lactose intolerance. Because even human is mammalian, I mean, babies used to be fed with the milk, and they don't have lactose intolerance. But with the age, we are losing this ability to produce the enzymes that can utilize lactose, and this can make the problems. Even who has a cat at home? Cats, no one has a cat, only few. Well, the cat as well is a lactose intolerant, and technically not supposed to give the milk to the adult cats. Well, they like it, but this could make them a stomach problem. Colon cancer, because of the interaction with other parts of the gastrointestinal place, interaction with the receptors, improving some metabolites, killing some of the cells, improving the immune response. All these things actually it's a points that are helping in the beneficial properties probiotic against them. Cholesterol lowering. Simply, they can destroy lactic acid bacteria can metabolize cholesterol, and it is a very simple way to reduce the cholesterol. Lowering blood pressure. You reduce the cholesterol, you can actually come as a consequence, you can reduce the blood pressure. Improving immune functions and preventing infections. Some of the products can directly fight with the pathogens, some can interact and induce the production of anti-inflammatory cytokines, but a very important depends on the situation. We was observing in some experiments that some probiotics, depends on the body temperature, they can act on different way. In the normal body temperature, they can be improving production of anti-inflammatory, but in high body temperature, they can actually be responsible for inducing production of pro-inflammatory. So again, it's coming another point that very often people don't pay attention. Probiotic maybe could be taken in the healthy condition, but not taken in when you have a high temperature, fiber, this hypothermic conditions. So again, something that merit more research because again, very often they develop the probiotic. They was looking for healthy people, and they say, well, for the healthy people, this can improve their conditions. But again, when you have the specific medical complications, maybe probiotic was not really appropriate. And in opposite, sometimes we need to think about, well, coming one step back. Probiotics are not medicine, they're supplements. But again, we can think about this probiotic as a supplement will be dedicated for people with some medical condition, some clinical conditions. So we need to be sure that in these clinical conditions, probiotic will be beneficial. And again, if it's possible to not be negative or neutral for the other patients, it's even better. But something that merit very interesting future research. Helicobacter pylori, this microorganism that is normally acting in very low pH in our stomach, that is a highly difficult to cure with antibiotics. It takes several months to cure if you manage, because sometimes the people having Helicobacter pylori for years, even for decades, and don't manage to go rid of them. Some probiotic and postbiotic, in fact, they're able to reduce the level, even to cure the Helicobacter pylori. There is a product from one company in the US offering a postbiotic, dead cells of Lactobacillus plantarum, and together with the probiotic that is Saccharomyces cerevisiae, they need to be taken for more than 3 months. But then this kind of product is able to cure the Helicobacter pylori. And my father is a good example of this. He was very strict, taking for almost half a year, and medical doctor was not able to detect any more Helicobacter pylori for him. And he tried several times an antibiotics treatment before, it was not really working. But this product worked. Well, I hope we'll work with other people as well. But again, the companies producing and selling it as these supplements for this purpose. So obviously, it's other questions was had as well. Antibiotic-associated diarrhea. Remember, you're taking antibiotic, you're killing everything in your gut. There's no any, well, the antibiotics, they're killing everything. They're not selective. And you don't have options because when you take, you have the disease, you need to take antibiotic. If not, you'll be dead. So very often it's happened that you coming to the point that you completely destroy your gut microbiota. So what you can do in this case, you can, very often the medical doctor, they suggest you to take fermented food products. In Bulgaria, will take telling to you, take antibiotic, take a yogurt, take more yogurt, take this drink, Bozan. Most probably here the doctor and your grandparents say take a daki all the time because it's a rich of lactic acid bacteria. So indirectly you're taking these beneficial microbes and trying to restore and to keep your gastrointestinal balance in the good health. These days, the medical doctors say, yes, you take a probiotic together with antibiotic, but now it's coming a problem. Probiotic don't have antibiotic-resistant genes because this is one of the rules to develop new program. Well, you're taking probiotic and antibiotic together, what will happen? Antibiotic will kill the probiotic. So sometimes they forgot to tell you to don't take it in the same time, take it in time difference. 3 or 6 hours after taking antibiotic, take a probiotic. So one cannot interfere with the other one. I was telling you in some of the previous lectures that it's a kind of movement that some of the probiotics can have reasonably acceptable antibiotic resistance. Just with this simple idea. If the antibiotic resistance is not on mobile elements, it's on the bacterial chromosome, and it needs to be studied to show that it's not going to be a good option for the transfer, this maybe it's not too bad idea, because in this way, you can take antibiotic together with a probiotic. So it's a kind of opinion, scientific opinion for some researchers that defend this idea that moderate antibiotic resistance, in fact, is not a wrong way to have a better option for treatment. Reducing inflammation. Several of the probiotics can be expressing different anti-inflammatory cytokines. And this way, they can actually reduce inflammation and will have their beneficial properties much more better. Improving mineral absorption. They can actually reduce some of the minerals because not all the minerals are good for us. They're heavy metals that we don't want. We have some other minerals to be absorbed because our body needs some of these minerals. They're essential for normal functionality. Preventing harmful bacterial growth under stress. Again, I sometimes am giving this example to the students in Sao Paulo. You can see at the university because during the exam, your immune system is going down. I mean, it's normal. You're under the stress, immune system going down. So during the exam, you can catch the disease. Go and say, because of your exam, I catch the disease. Good lawyer can help you. It's a job. But then it's interesting because we're full of stress. I mean, you're driving, you are traveling, you're flying, you're taking bus, train. You need to figure out the job, accommodation, everything. It's a stress. Our life is full of stress. And during the stress condition, we actually having a much more, our immune system is much more going down. And different pathogens can make a profit of this. And the probiotics with the normal interaction with these pathogens can actually reduce their effect. Irritable bowel syndrome and colitis, another example. Several of probiotics was developed to reduce these conditions. There are several probiotics that develop and on the market improving allergic reactions. Very often they can actually interact and they can destroy, they can metabolize some of the allergens. But in this way, they actually helping to improve all. Now, traditional fermented food products are their extremely good example how we can improve and we can use these fermented food products as an extremely good source of probiotic bacteria. Good point. Very often, these fermented food products they're already known by traditional medicine. Well, if we know that specific fermented food products is good for something, traditional medicine is recommending, why not explore and to see what's the reason? Could be some metabolites, could be some phenolic compounds, could be some extremely high levels of vitamins, but could be microorganisms that are in these products and metabolites produced by this microorganism responsible for these beneficial properties. So we was having this project isolating the different lactic acid bacteria from kefir, molasses, olive grain as a potential source of beneficial microorganism. Well, in the first stage, we actually isolated as a bacteriocin producer, but then we wanted to see and explore their potential probiotics properties. Well, all of these strains isolated from this fermented food products, I remember I was telling you, when I'm giving name, I'm putting some initials in the end showing the origin. So KF is kefir, MS is molasses. This one is from olives, but because we don't want it a symbol O, because very easily can be confused by zero. You know, it just make more fat or more thin, it means zero or O. Fat is O, thin is zero. But again, when you have printed text, sometimes it's difficult. So my boss insisted to put his initials here. So he wanted to build the name of the strain as well. And 423 is a plantarum that was already talking about yesterday. So this strain was showing an interesting profile in antimicrobial profile. So there was already we know they're producing bacteriocins, and some of them was with a very good activities against listeria. But then we wanted to see what a story about the antibiotic resistance. So you see, we was not really respecting EFSA list, but we have all the antibiotics recommended by EFSA in this list because it's important. We have even more. And then we will see that they're not too bad regarding the antibiotic resistance. So we want to see addition to the CACO2 cells. Well, you can say, but you're always defending that CACO2 is a cancer cells. But additions to the CACO2 could be easily transmitted to other cells. Because the models showing that when it's a question to the addition, CACO can be used as a model for the healthy cells. It's not a big problem. But if you look for the expression of some cytokines, CACO2 is not really the best option to study expression of the cytokines by the CACO2 in some specific conditions. Well, we have a different, obviously different additions. But again, remember, we don't want all the probiotics to have good additions. We need to know how they adhere. But again, it's not compulsory. And as well, we was checking some markers. Because you have some genes that are responsible for the additions. There's a MAP, MOOP, elongation factor, and then some other genes. And easily by the PCR, you can detect. Interestingly, most of these genes been described for the Lactobacillus plantarum. But again, we was having even other species here. We had Enterococcus, Leuconostoc in this case. And interestingly, we detected these genes in the other. So now it's coming the question. Sometimes when you perform the PCR, you're looking for the specific gene for the specific species that you work for. But sometimes just go a little bit broader. We'll be surprised to find that some genes that are not described in some species could be present. And this is interesting point to observe. By the way, this work is done with one of my collaborators in South Africa, Kamini Ramaya. So the name is showing what's the origin? Indian. And she was very, very nice girl because always in the perfect mood, never been in the bad mood. Even if it's happened the worst thing in the life, she still will be in the smile and try to be a positive and find a way. Because we were struggling a lot when we needed to do the RNA work with these strains. To isolate the RNA, to do the expression of these genes, not always working well, but we managed to, because she was studying the expression of these addition genes in Lactobacillus plantarum 423, the strain, to see how the strain can be really acting as a probiotic. But then we decide, okay, then implement all this work that you already done with Plantaro to the other strains. And we were showing that the genes was present, and that was actually expressing them, not only present, but that was actively expressing. And depends on the condition that you have the strain, you can have modulation of this expression. In this case, it's very important when we do the real-time PCR to check the expression on the RNA level when you have the strain in the condition when it's supposed to perform his duty. Because very often you can be really surprised that the strain expressing some beneficial metabolites only when it's in the gastrointestinal condition. In the normal MRS, they don't want to express, but when they go to the gut condition, we can modulate this and we can simulate, you can detect this production. Um, Professor Tivari, can I have a little bit more example and then we have a break? Okay. Then we was using this boza. Remember this, I was giving to this example before, as a vector for delivery of probiotic lactic bacteria to the cough. In this project, we already know that the strain Enterococcus multi producing antibacterial and antiviral peptides. We know that having a very strong activity against different pathogens, we know that the strain is a very good probiotic because we even made the experiments in the animal. And very interesting, in one day that company come and say, but listen, we feed our mice with this strain, but you know they're not getting fat. They just build on muscle. Because it's stimulating even muscle production. I say, okay, but why not suggest this to the farmers that do the pig farming and they can pig with more muscle, not with the fat, because when you buy the meat, you don't want the fat, you want the meat. And then there was actually a different project going as supplements, probiotic supplements for the farming animals with the idea to build more meat and more fat. So there was even showing pro-inflammatory, anti-inflammatory properties when the animals was receiving this strain, improving the immune response, improving even fertility in the female animals. So it's really complex work showing the beneficial properties in this strain. Strain was having a very interesting activity against some gram-negative Klebsiella. Some strains being sensitive to the strains, some Escherichia coli, some Clostridium, some MRSA, Staphylococcus aureus not showing sensitivity. We checked just two. Proteus mirabilis, Pseudomonas, not all. We checked 19, 6 only was sensitive to this bactericide. But again, this bactericide was very potent, and as well the strain applied as a probiotic was having a very good idea. Strain was having a reasonably good growth when exposed to the different percentage of ox bile, and this is important because the strain can survive in our gastrointestinal. And as well in different pH was having a good growth. Well, Enterococci normally they don't complain. They can grow in the pH in ox bile. It's a part of their nature. If you have Enterococcus that can be fully killed by ox bile is something new. You're going to have a good paper in a good journal because Enterococci normally they survive ox bile. But the opposite will be a very new species. So we wanted to see a gradation property of this strain with a different beneficial pathogen strains. So we have some beneficial, some pathogen strains in this work, and it was showing aggregation because remember, bactericide needs to get together with the pathogen in order to kill it. If they cannot attach, it will be very difficult to kill. So aggregation properties are very important preliminary test in order to know if the strain will have a good behavior and good applications program. As well, we was checking this absorption of the bactericides to the different organisms, to positive, sensitive and resistant to the bactericide, to see to predict if some specific pathogens will be more or less sensitive. Do you have curiosity how this test is going to be done? Do you want to shortly to explain now? We want it on the discussion now. Okay, let's do it now. You're having a cell suspension of the test organism that you want to explore. You're having your cell-free supernatant of your bactericide. So you just need to know how many arbitrary units do you have of your cell-free supernatant. How much is the bacteriocin strong there? Then you mix bacteriocin together with the cell suspension of the test microorganism. You expose them, let's say, for 1 hour. You centrifuge, you need to correct the pH again, and then you do again arbitrary units. And you compare arbitrary units in the beginning, arbitrary units in the end, and by formula, that is on the papers already published several times, you can calculate this percentage. Very important tip, if you do this test, remember, if your bacteriocin is very strong, you need to dilute. Because something like 12,000 units is a good level. 6,000, 12,000 is a good level. Because if you have a very high activity like 100,000 or 50,000, you're not going to see the difference. Because some bacteriocin will get absorbed on the surface, but compared with the leftover, you cannot see the difference. So you need to dilute your bacteriocin to like 12,000 units, and then you can do the test and you can see this difference in calculating the absorption. We wanted as well to see how the temperature interferes with this absorption. Because look here, you have absorption to Listeria, to Enterococcus faecalis. A very good absorption is in the focus 25 to 40 to 37, more or less here. Less around. So it means the strain having a good potential to be applied in the human being, in 37. Or even in some fishes, because, but if you want to apply this bacteriocin because of this absorption, because you're applying the strain will produce the bacteriocin. Obviously, in the cold storage food products, not going to work such a good like in the human. So from this point of view, this simple method to do, you do the absorption study at different temperature and in different pH. You see, in neutral, it's working much more better. So probiotic will work much more better in lower part of our gastrointestinal part. Obviously, in the stomach, no way. Not going to work very well. Most probably against Helicobacter pylori, it's not going to have a good chance. But down in the lower part of the gastrointestinal part, when the pH is like 6, 7, we'll have the good option to perform well. Different chemicals, we check the absorption in the presence of different chemicals. Some of them are clearly from academic point of interest, some of them from the practical point of interest. But again, you can think about, okay, I have the medication together with our probiotic. There are some chemicals there. I can have the food products and enrich my gut environment with some specific protein, dairy protein or plant protein, and you can actually explore and see if this will give some interference on the absorption process. There was having a very good bacteriocin cells, very good activity, healing very well enterococcus, less little bit recovery of Listeria innocua. But again, this is some points that again will give you indication if can be a good option to cure some of the foodborne pathogens. As well, when we was exploring the percentage of the cell lysis, you see even 20 units and 12,000 units, they was reasonably equal. So you don't need to overload with the bacteriocin. And it's another important point. When you apply the bacteriocin, do you think they will produce in the same way in our gut like in the MRS? No. MRS is a perfect media, and very often they can express very well in the perfect media and much more reduced level when they in our gut. But there was several works showing that in fact several bacteriocins can really express in the gut environment. There was a work showing and proving this. So, but again, even 200 units, that is very low activity. It's enough to perform well against Listeria. Going to a different way, we was showing by the different approaches that we can use the reading of 260 or 420. Remember, here was a DNA, RNA proteins, liberation from the cells, presence of the beta-galactosidase as a fact that it's an intercellular enzyme, and if you treat the cells with the bacteriocin, you destroy them and you liberate all these proteins. We're showing that, in fact, this bacteriocin was doing well. But there is coming question again, antibiotics. We needed to check. You see a long list of antibiotics, but most of them are from the same groups. And then we was having, including the different antibiotics as well, to be sure about the safety. But again, as I say, some of the antibiotics, we believe that some moderate resistance could be actually a good point. But coming a different point, what about the other medications? Normally, if you are in the perfect health, you're not going to take a probiotics, right? If you start having a problems, you're looking for them. And very often you take another medicines, and especially we need to pay attention to the medicine that the people taking for rest of their life. If you have, let's say, diabetic, you have the cardiovascular diseases, you need to take this medicine every single morning, no option. And very often these medicines, these pharmaceutical products, they can interfere with the probiotics. There's another point that merit more attention because medical doctors very often don't telling you this. And as we discussed in the beginning, no one read this huge paper inside the box. If you're taking, let's say, ibuprofen, it's an anti-inflammatory. Ibuprofen, it's a group of the sodium or potassium diclofenac. I mean, it's quite popular medicine in anti-inflammatory, but the problem they have negative effect on the probiotics, they're killing probiotics. If you read this paper inside the pharmaceutical product, it's written, don't take it for too long because it can have negative effect on your gastrointestinal microbiota. But again, people don't read this. This is important. You need to educate the people and explain. Please read it. I mean, it will take you a few minutes to read this, and then you know for some possible negative effects. But several probiotics could be killed by this anti-inflammatory. Some other medications, like even the neuroleptic trioridazine or dioritidine, that is cardiovascular, they was having this killing effect against probiotics. And now you need to do the minimum inhibition concentration because only by this way you can see if they will have the really negative effect or not. Because if the minimum inhibition concentration is very low, so you need a little bit to kill the probiotic, then you're in trouble because you will take this medication like every, say, 8 hours, every 12 hours, once per day, and normally you're reaching quite high level in your blood plasma of this. And since it's in the blood plasma, this can be transmitted to other environment in the body. And then imagine if you have at this level, high level in the blood plasma, how much is in your gut, even more. So this is important to be sure that medication that you take is not going to interfere with the probiotics. Again, most research is needed in this area because not sufficient information. And even he was having him in there to generate a website, free website, where we can upload all this data by anyone in the world that was doing this kind of work, so the medical doctors can have this access, because sometimes they don't know. They don't know if this can interfere or not, but something that merits additional attention. So it's more or less like that. Imagine you're taking every 8 hours your medication. Your level of this medication will go in this way, more or less, in your body with some fluctuation. And if the minimum inhibition concentration that will kill your probiotic is, let's say, 75 units, obviously you have much more of your probiotic, of your medication in your stomach. This probiotic is not going to work. You will be killed. If you need, let's say, 200 and 150 units of your medication to kill the probiotic, I mean, the level is always like that, so this probiotic will be happy to survive again with medication. For this B, we don't know. It's because it's fluctuation around, so most probably could be a problem as well. Then we wanted as well to see if we can produce and kill inside the product. So we made it fermented milk, mixed culture of our producer and our test organism. It was doing a level of pH, how the pH decrease, how the blue is a total cell count. This one is the listeria cell count, and the level of bacteriocin that we can detect. Obviously, we detected much more low activity, only 50,000 units of our bacteriocin in this condition. but as well compared with this is compared with the MRS. But again, it was enough to reduce the level of listeria in this system. But then remember, I was telling you that addition of the probiotics can depend of the to the cell line can depend on several components. So we was doing addition to the CACO2 cells, but now taking consideration normal CACO2 cells with our probiotic in presence of periodate, trypsin, pronase, pepsin, because different proteolytic enzymes will be present in our gut. We was doing all this process in presence of oxbal and pancreatin. You see, addition was very bad when oxbal was presented. So the presence of oxbal can actually reduce drastically addition of our probiotic to the CACO2, but some other components was not really interfering significantly with this idea. And this we can go in the next after the break, because I'm sure you want to have already tea and snack. And since you survived your end, chocolates. Okay, how are we going to do it?
After the lecture.
After? I was thinking, no, on the tea break. Okay. Where's the tea break? That's where. Okay, let's start here with the chocolate. Can you help me, please? Thank you as well. You know what? No, it's okay, but for you as well. Just go for it and then finish all of it. Okay, enjoy. Look at the coffee and enjoy it. No, it's not. Well, photo ले लेना है. You think you have a photo? I think that's in that. Okay. Yeah, different. I got a date, so not anymore. Thank you. This is all this.
just want to make a question. I have the curiosity, who took the yellow, big one yellow?
Did you get that one?
Yes.
The one
Normally in Brazil, they say,
she got the yellow one.
yeah, I think, no, that this is the small yellow. The yellow, the round one.
This one?
Yes. Normally, the girl's giving to the boys as a declaration of love. Not normally it's like that, but well, it's just a joke, don't worry. So you go home, give to your husband, then everything's okay.
Can everyone had the chocolate, right? So, was enough? Okay. Well, it's not enough. I have more in the room. Can bring more. Okay, let's. Now it's working. Right. Okay, why is so dark the screen? Lamp dimming. Okay, right. There's something I can do on the computer or not? Okay. Well, we continue with the story with again. Remember, I was showing you this example in the previous lecture, but now we're looking from different perspectives. Now we don't want to kill them. We don't want to kill the test organism. We don't looking for the food safety. You see, the same experiments can be applied in the food safety, could be applied as a probiotics experiment. Because we're producing boza, we're making all this process, we inoculate, remember, with the boza, with the test organism. We wanted to know what is going there. And they're growing quite acceptably. You have the good population numbers of the microorganisms. And then you're having the DG. You want to see, in this case, I don't care about disappearing of Lactobacillus acti because it's not my objective to have disappearing of the test microorganism. My objective in this case is to have Enterococcus multi present in the system. And the strain is present because was confirmed by the normal species specific PCR, and this will be where is here. This PCR confirming that Enterococcus multi with the species specific primate is present in the product. So starter culture Enterococcus multi, making the Enterococcus multi to be present in the process. And in this way, we guarantee that our product, fermented food products, can carry the probiotic strain. Again, analysis of the organoleptic properties was clearly showing that the consumers cannot detect the presence of the strain. And this is extremely important when we develop the new fermented food products, because if you change the organoleptic properties, people don't buy it. They don't want it to have it. And you need to make them happy in order to provide these strains and these products to them. And he was using actually GIT model. Well, not only in the literature you saw already, Scheim, Team, etc. We have this model in South Africa we call Frankenschwein. In German, Schwein it's a pig, Frankenstein, Frankenpig. And interestingly, was easily working. You can see you can easily make this gastrointestinal model in your lab. You need a chamber to put all these things because it needs to be an appropriate temperature. You need some pumps. You move from one another part of the stomach. You're adding some gastrointestinal fluids. You can take a sample to have the pH meter inside to change to correct the pH, etc. And you can do it relatively easy. It's a dynamic model. However, it's very simple, but again, it's working. Now we're developing the new model in our laboratory that we can actually easily follow the changes in the microbiota. We're going to inoculate our models with appropriate gastrointestinal microbiota. But the important thing, we need to pay attention because imagine if you have milk here, it's okay. It's working perfect. But imagine if you put the grains, if you put the corn going to go with these pumps, no way. So we actually modifying our toast in order that even the pieces of the food can go via the system and could be applied. and hope in the next couple of months we'll be finalizing and are already running our first experiments. This is together with our collaborator in Russia and doing this kind of work. Even there's some models now, they actually was linked together with the sequencer. So automatically they can take material from the model, they can do even genetic sequence, they can do metabolomic, everything automatic. But where's the fun? I don't want a model that you put something in the front and you wait 1 week to have the results. I want to have my hands there and to know, to have the control on this process, to enjoy all this work. We have another kimchi project, that's called Kimchi Project, where we was exploring the different probiotic properties of kimchi. In this kind of work, we wanted to know, to develop actually some alternatives. So we was looking for the property strains in order to select the good strains for the developing of new therapeutic products. If you look here, the antibiotic discovery resistance timeline, when the antibiotic being discovered already 12 years later, you had a resistance. Then you have more antibiotics and again resistance. These days we can have some alternative. We can have several bacteriaceae listed here, and they can be applied in a different process for resistance to different diseases. I mean, we can have the area of application, vancomycin resistance, insulin resistance, Staphylococcus aureus. We already have application of niacin and lactacin for treatment of some RSA or vancomycin resistance. In case of treatment of Clostridium difficile, we have multicin, niacin as well applied, generally multicin. All these dental care, contraceptive, skin care, tuberculosis, antiviral, antifungal, different pharmaceuticals, different bacteriaceae and with different applications. So it's showing that in fact bacteriaceae can be considered as a next stage to kind of at least temporary win this battle against antibiotic resistance, because we needed more and more way to do this kind of treatment. In this kimchi project, actually we was isolating different microorganisms from the kimchi. You see how they're producing kimchi. They need to treat, you know already the recipe, or you just know the product? Okay. They needed to incorporate these spices, red chili and salt and everything, but again, we was trying to isolate microorganisms, and actually was quite a lot of microbes there. It's a really living system. They're having jobs. Sometimes the students go home, and they're going for the kind of holidays, whatever. They're coming back after that, and they taking kimchi with them back from home to the university. Very often they carry kimchi in the plastic bag, and the kimchi is fermenting even in the tray. And sometimes bag can break, and you can smell it very well even in the tray. That's the one carrying kimchi. So don't be so positively about the kimchi. Most of the kimchi have a very good taste, rich of several beneficial microorganisms. But since the kimchi is a cabbage, right? And the cabbage is growing where? On the ground, no way. And very often can be contamination with bacillus. Even we detected in some samples, even Bacillus cereus. And Bacillus cereus can be a very nasty foodborne pathogen. So, in fact, very important hygienic condition in the production of the kimchi needs to be applied. And you can believe me, not always people manipulating work like that. Sometimes no appropriate clothes, no gloves, no even appropriate containers to do. So it's important to have the appropriate hygienic conditions. So isolated some of the bacteria, we have production of the bacteriaceae, and then we was doing a red PCR to prove that we have the different strains. And we have two different strains, Enterococcus faecium and Enterococcus 120 KC. Well, we see ST initials, 20 number of the isolates, KC kimchi. and then we had isolates 41. And these two strains was showing very good survival in the part of the gastrointestinal condition. So when they were splicing the gastrointestinal condition, you have the time 0, time 1, time 2. Time 0 is like what's going in the mouth. Time 1 is considered when they stay in the stomach. And in the large intestinal, it's time 2. So you see in the case of strain 20KC, the first strain, you're having decline, but still, okay, look, it's decline, but this is 8.10 to 8, 4.10 to 8. So sometimes you need to see very carefully the scale because it's declining just even not one log. So it's a good survival. It's again, if you look here, it's a look big, but you need to look the scale on the side. What is it? And at the same time, Enterococcus fecalis, the probiotic strain from the laboratory, was having more or less the same behavior. So the strain was having a good potential, these two strains to be applied. And then we wanted to see how, when we do the combined culture, because not only to have the probiotic strain, but we wanted to see when the probiotic strain is co-culture with the Listeria monocytogenes, because we know the strain having a negative effect, inhibitory effect against Listeria. But in this case, we culture together, bacteriocin producer together with the Listeria. We see the good survival of the bacteriocin producer, but practically for almost 100% remove of Listeria from the sample, from the environment. So if you have the patient that having Listeria contamination and taking this probiotic, in fact, will help to not only to remove Listeria, but prevent of future Listeria contaminations. For the people that are in the higher risk, in fact, this kind of probiotic will be perfect because it will be helping in prevention of the Listeria contamination. Well, the best thing, if you are pregnant, please don't eat the food that is a high risk. Because Listeria monocytogenes could be killing not only your future child, but can kill yourself as well. Don't give it to small children. Don't give it to the elderly people. Don't give to the people with the immune response problem. Now, when you're talking about immune compromise, what is coming in your head as a first option? Which people are immunocompromised? Come on, answer. Can I put in the test this question? Well, normally the people always will say HIV. They're immunocompromised. But they're just a small part of the immunocompromised people. If you have organ transplant, you're immunocompromised because after the transplantation of the organ, you need to take for quite a long time immunocompromised medications. If you're in some chirurgy, you're in the stress, you're immunocompromised. For all these people are actually as well, they're immunocompromised. Even as I say, you're making exam, your immune system is going down. So all the pathogens will take advantage and try to make your life more complicated. So we made it as well the metabolomic analysis because we wanted to see what kind of metabolites are produced by these strains, because obviously not only bacteriocins, and unfortunately it's not in the color, but darkest color, normally they have more present. So you see several acids was increasing in these strain environments. So not only bacteriocins, but the acids and even some additional antimicrobial metabolites can be detected. And that was improving the antimicrobial effect of when these two strains was growing. So in this case, effect of different enterocins produced by different strains of Enterococcus species, obviously they have this very strong effect. And we was actually selecting for future study a different strain, Enterococcus faecium 7119, isolated from, this is the initials from one fermented soybean pest in Korea. And we wanted to see again how they would. So we treat the vancomycin-resistant Enterococcus faecium with our bacteriocin, doing the flow cytometry to to follow what's going. You see you have live, damaged, and dead cells. In most of the cases, this is the control, and different level of the bactericide when applied. You have a lot of dead cells and a very small amount of damaged cells when you apply 12,800. When you go further, you have kind of breaking point between 6,000 and 3,000 units. So now we have live cells, dead and damaged cells. So obviously to be effective this treatment, you need to have something a little bit more than 3,200 units. Because if you apply less, most probably you will actually not kill. You can have several damaged cells, and these damaged cells actually later could be maybe recovered and make you problem. If you use much less bactericide, you have several live cells and little bit of dead cells. So minimum inhibition concentration is somewhere between 3,000 and 6,000, and you need to apply it again in enough concentration in order to be sure that you're going to reduce and you eliminate, not just to making sleep this vancomycin system, but really kill it and remove it from the system. So we was using again to explore the beneficial properties of symbiotics between the bactericins and some antibiotics. In this case, we was working against vancomycin resistant to kill the vancomycin resistant enterococcus, and we was applying combination of our bactericin and vancomycin. So you can see in these two cases with the two different. This is bactericin 1, bactericin 2, bactericin 1, bactericin 2. We was showing that in fact, when we apply the vancomycin, they're not supposed to work because they're resistant. But you apply it together with the bactericin, they work in combination. And even you can reduce the quantity of antibiotics. An antibiotic that is already been a problem, it's not working because of resistance of the test microorganism. Together with the bactericin, you can have the effect and you can have the beneficial properties. As well, the ciprofloxacin, use it as a control. We was observing the same pattern and again, interaction between bactericin. Again, doing the same experiment with the live dead cells, we was observing that in most of the cases, dead cells and not live, not just damaged, and effectively working. Then we wanted to analyze this data, and we was performing, because you're having kind of mathematical models that can tell if it's synergistic, is it just the additive effect, indifference, or antagonism? Depends on the level of these factors, 0.05, 0.5 to 1, below 2, or between 2 and 4. And when I was performing these experiments, in fact, different concentration of the antibiotic and the bactericin, these indexes, in fact, was showing that in most of the cases we was having synergistic effect. So it's confirming that we can apply the allobactericin together with okay, it's almost 10 to 12. We was using fish roe application as another, and then after finishing with this example, we can actually go for the lunch break. We was exploring the use of fluorescent in situ hybridization together with flow cytometry. We wanted to combine this method because we was having this idea. Normally the people do a fluorescent in fish application when you use the specific fluorescent marker that where they will attach to the cells, and based on the fluorescence you can know what do you have. Well, but this image was telling you something. It's telling you, yeah, I have live, I have dead cells, I have a cell that I'm targeting, but how much? It's a little bit kind of, okay, it's a nice image, but not really giving you a significance of this treatment, because you're having the cells, but they are too much, they are less, they are acceptable, they are non-acceptable. And actually, the idea is to combine the fluorescent in situ together with the flow cytometry, and then you can make a quantification. How the process more or less works? You need to select and design your probes. So you need to have specific probe that will give the fluorescence. And I just, please, the girls from the lab of Santos, remind me to generate PDF today on the lunchtime and to put it on the system. Because I always forgot. I can do it in this computer in the end. I think it's easier even. So then You design your crop. It's not a really big problem. This has several computer programs these days. It's quite easy. Then you need to select the way of the samples, and you need to perform first hybridization. So you need to find appropriate conditions because you play with the temperature, you play with the time. So to have a really targeting, specific targeting of your crop with the DNA of your organisms. So you have the samples, you make fixing, you put your marker, and then after that, you're doing the hybridization, you need to do visualization. You can do it with microscope, but again, it's a little bit complicated. So after the fixed and permeabilized cells, you having this process of the putting your target, your marker where you want. You made the contact with between the ribosomes and your marker, and you can observe. In this case, we're going a little bit further. So we're doing the samples, we're preparing, we hybridizing, we're doing in the appropriate concentration in order to work. And then we put our fluorescent marker, doing incubation, and then going to, okay. Well, I was supposed to click to go to those steps. And then you're going to the flow cytometry when actually you can analyze. In this work, we were taking to want to validate our idea because we made it in the pure culture. So we took the different samples from the patients from bacterial infections. Here we do the classical microbiology to see what we have there, what we don't have. But then we was performing the analysis, and we selected the appropriate vancomycin resistant strains. We was isolating the strain that are purely vancomycin. So we wanted to detect vancomycin A and vancomycin B genes. By the plate, we know that our bacterium is working against this vancomycin resistance. And when we do the fish flow cytometry, in fact, we was able, okay, we was able to detect much more quicker by flow cytometry, because if you do the isolation, if you do the test, you do the antibiotic resistance, you try to do, it's taking a long time. But with the flow cytometry method, you have the marker for the vancomycin, you have the marker for the vancomycin B, so you actually make the probe, they detect, they catch these genes, and with the flow cytometry you can analyze. So in this case, our strain, Pediococcus pentosacellus, Proterosin producer, was able to inhibit vancomycin resistance and to able to kill it. So with a very small amount, we was able to perform this work. We was showing even interaction but when we was doing this flow cytometry, we was able to detect the fact of the presence of the vancomycin resistant genes in this total isolates. So if you work in the classical microbiology, you isolating a strain, you trace this strain. If you do the fish flow, actually you can take a total sample, you have the probe that you already validated, they recognize the vancomycin gene, they was making contact, they do the fluorescence, and by flow cytometry you can analyze and you can say, yes, our sample is carrying vancomycin resistant genes. And obviously you're not supposed to use the vancomycin for treatment of these patients. So we get even further. We developed the probes for the different antibiotic resistance, not only the vancomycin. So we was performing with the pure culture first. We know with that we can treat them. And after that, we was doing the two. You're taking sample from the patient, total samples, fecal samples can be even. And then you do the total DNA extraction. You do the treatment with these probes for the different antibiotics. You do the hybridization, that is about 1 hour. And then the flow cytometry takes about 15 minutes, and you can see something like that, and you can see the patient has presence of antibiotic-resistant genes. You're not going to give this antibiotic, you select the antibiotic that is more appropriate. So all this process can take less than 1 day. If you do the classical way, it will take you a week. In this way, patient coming to the hospital in the morning, in the evening, you already know what kind of antibiotics you can apply and how you can apply it in order to be effective. Can we talk about postbiotic a little bit? Then have a little bit of this. Let's have a discussion and put the postbiotic in the after lunches, okay?
Yes, yes.
Because they informed me that I need to go for another meeting at 12:30. So let's do the discussion, and postbiotic we can do it in the afternoon session.
Oh, no problem.
So what's your curiosity now about this part? Do you think probiotic could be really a good idea when we need to be careful? In my opinion, probiotic can have a very good application. I mean, they show it, like almost 1 century, we apply them in one or another form. The problem in the beginning, we don't know too much about them, and everything look much more easy. In these days, we go deeper and deeper, and we realize we need more and more study. You remember, there's a story from the old Greek time. Socrates, the philosopher, he was used to have his lecture with his students on the beach. Well, not too bad idea. Well, Greece is the peninsula, the coast is nearby. And one day the students, they asked Socrates, but you know everything. Every time we ask you something, you have the solution, you know everything. He was taking one stick and draw one circle on the sand, circle, and saying to the students, listen, this circle representing your knowledge. And here draw another bigger circle around and say, this bigger circle representing my knowledge. Obviously, I know a little bit more than you. But now think about something else. This line around the small circle, how long is it? Representing things that you don't know. And now think about the long line around my circle, representing things that I don't know. So you see actually the beauty of the science, you go and discover more, and you realize in fact that it's more things to be discovered. And it's actually making it interesting, because you're not satisfied what you already discovered, what you already know. You want to know more, and more, and more, and more. So this is actually the interesting same thing with the probiotic. In the beginning, we don't know too much about them, but we don't know what we must not even imagine what we don't know. So with going more deeper into the area of the probiotics, we realize that it's actually the unknown is much more bigger. And this is actually making space for everyone to work in the area and to try to discover these answers. So I know someone also several times mentioned that work with the probiotics. And yeah, so what is your challenge in the lab normally? Feel free to share what is the problems, what do you think needs to be done. You have some ideas. You see, in the beginning, people was thinking that the fermented food products could be a good option. Even with several fermented food products was marketed as a probiotic in the supermarkets, because the word was selling almost everything. But then they realized the regulations and main regulation bodies, they say you cannot claim as a probiotic if you don't have sufficient clinical evidences for this. So they start to remove the name of these fermented food products and call them beneficial products, functional products, but cannot call them a probiotic because very often they don't adjust to the definition, because you need to know what strain do you have inside. You need to have the clinical evidences, how this strain is working, why it's working. And as well need to know the potential negative effects. You don't want to put the probiotic products on the market that they have too many negative effects. Or if there are some, you need to aware the customers. So this probiotic is not good for you because you have diabetes, you have the high blood pressure, it's good for your mood. I mean, some beneficial metabolites like GABA, they're very beneficial for the nerve transmissions. But again, that was interesting because they could be, GABA is a very good option, but again, too much GABA, it's good. Maybe not. Maybe it needs to be a right dose. I mean, Paracelsus was telling that the poison, it's a dose dependent. Something that in the high dose maybe it's a poison, in low dose it's a beneficial. Homeopathic, it's the same thing. Some small amount of the metabolites, they can stimulate the immune system. So now the homeopathy is a probiotic? No, because it's not live microorganism, it's some metabolites. But again, can we maybe call the postbiotic homeopathic products? Maybe, why not? Because you have the metabolites, you provide some metabolites in a certain level, and you stimulate the immune system, you have the beneficial properties. When we go to the postbiotic story, you can realize that, in fact, several of the probiotics benefits, they are realized on the some metabolites produced by microorganisms. And these metabolites can be even provided without live cells. They don't need to be alive. They are good because they can be produced during the lifespan. And if you have the cells that is colonizing the gastrointestinal part, they can actually produce constantly these metabolites. But then is coming a problem. What about immunocompromised people? This microbiota there, it's a good or it's a problem. So maybe the application of some semi-purified or some even crude extract, but not a live cell, could be a good option. You see the questions start coming and coming because when Mechnikov and Stam and Grigorov, they claimed that yogurt and Lactobacillus bulgaricus is good, was simple. There was not metabolomic, not genomic, no proteomic. It's not all this study. But in fact, the development of the technical advances in the science was helping to make a research and to answer the questions more complex and in shorter time to perform the experiments that will give the idea about the benefits, about the safety, about the danger of these microorganisms. No questions, no comments, more chocolates. I think someone wants to ask in the first row. It's a shy but one.
No. So actually you discussed that bile reduces the adhesion property. So what could be the reason behind this?
Of bile.
Yeah.
Well, this could be changed in the presence of bile. Most probably could be changed on the surface. Maybe change of the electrical charge. Maybe it's just a simple interaction. Maybe it's addition of some other components is better. But again, think about biles where they are. They're more in the upper gut. No, right after the stomach. What's the role of the biles? They need to reduce the cholesterol. They need to perform in the metabolism. If you go further in the way of the gastrointestinal part, they reduce it. So, in fact, this addition, low addition in the presence of bile, will give the opportunity the probiotic to adhere to another part of the gastrointestinal part. Sometimes you don't want them to adhere to one to another part. You want them to adhere to the place they can perform their work. Because if they're producing vitamins in the mouth, the vitamin is not going to reach place down when it's supposed to be. So you want a probiotic to perform his action in the right place. Again, you can do it by encapsulation this way. You can keep it probiotic with specific capsules, so it can be resolved in the place you want probiotic to be acting. Or sometimes the natural interaction in our body was actually preventing the probiotic to be better in one or another place. Actually, the nature was already doing the job. They created the human in the way. And sometimes we just need to be careful with touching this because it's a very delicate system. If you touch something that is making some changes, you maybe will regret later. The same thing, when the people in the beginning of 20th century, they was using radioactive water for some treatment because they observed that have some short-term benefits. But no one think about mutagenesis that is actually performed by this radioactive environment. There was several radioactive, mineral water in general could be a little bit higher radioactivity because it's coming from the deeper parts of the earth. And in some situation, they was recommending this water for some rheumatic or other diseases. To do the bad with this water, but then you need to put on the balance. What about the negative effect of this radioactivity? In the beginning of 20th century, they was not aware of this. This day we know much more better that radioactivity is not a good option. But again, every medicament, don't exist the medication, the pharmaceutical product that is only beneficial. You helping the patient on one side, but you giving a problem on another side. So the benefits needs to be more than the damage. If we have disease, we don't have option. We need to take antibiotics. We will destroy our gastrointestinal microbiota, but this is coming the probiotics and fermented beneficial foods to restore the microbiota. Again, yesterday, I think it was yesterday, we discussed in the afternoon session about the fecal transfer.
Yes, sir.
So it's a way to restore the microbiota after Clostridium difficile infections. Because of the high level of antibiotic treatment, you're going to have this reduction of the normal microbiota. Clostridium survive and is taking over and performing the product. Fecal transplant is one of the options. It's been applied for a long time, but in the Western medicine they started recently to apply because it's just need to be sure that you have the donor with a healthy status. Because you don't want to introduce another disease.
Yeah.
Poor person have Clostridium difficile, and now we're going to introduce Salmonella. Obviously, it's not a good idea. Any comments? Yeah.
Sir, do probiotics have any specific level of acidity tolerance and story and what else?
So it's to come in the front.
Sir, I want to ask, do probiotics have any specific level of acidity tolerance or it is totally dependent on what type of probiotic we are going
If I understand well, it's about the acidity.
to use? Acidity.
Yeah. Probiotics, they are bacterial culture. So obviously they will have their personal preferences of what pH. If you have lactobacilli, they are much more happy with the 3.5, about 4 pH. They can survive even a little bit lower, but enterococci was acidification of enterococci going to 4 point something, but they can have better survival in the lower pH. You see the growth condition and the survival, they are not in parallel, sometimes they're different things. Even salmonella, you can kill it with pH 4, but important is when you have the probiotic, you need to be sure that the probiotic will survive the acid conditions since he needs to go via the stomach. And in the stomach is like 2. Most of them, when they select, to do pre-selection, you look for this one that can survive. Because from the technological point of view, it's much more easy to give the pure bacterial culture. If you need to encapsulate, to incorporate to something, or make a specific pharmaceutical formula, it's giving more help. More simple, it's more easy. But if you have a strain that have a very good properties, a very strong immune regulations, very strong antimicrobial properties, very strong production of neurotransmitter, etc., you need and he don't survive, you need to go to these additional methods and to be sure that you can deliver in the right place and in the right conditions. Because technically, our body was built to protect. If you think about any bacteria that will come in our mouth, we don't want this bacteria to go down from the safety point of view. We have different enzymes in our mouth, we have saliva, we have beneficial microbes. There are more than 400 different species. They will try their best to kill any new bacteria. Then things going down. In the stomach, very acid condition, they will try to kill anyone because nature want to protect us. They made all these barriers in order to protect what's going in our gut. Then it's passing after the stomach, you have bile salts. They will try to kill us well. So you see, probiotic, it's a good guy, but nature was already create us to protect from any microbial invasion. So in this case, we need to select the strain that is a good survival from one side, or if we cannot survive, we need to help this strain because we're now talking about the additional, not normal, this is extra thing. Probiotic is not something that is part of our body. We're putting these things. We're selecting one from our body that is already beneficial, and we're putting in another person's. So we need to find a way how to protect them, to be ordered that they arrive alive in the place where we want them to perform. But then it's maybe one more philosophical point. If the nature, the evolution created humans in this way, why do we believe that we can change all these things in a week or 2 weeks? It's a little bit. Okay, if you believe in new religions, if the creation you by your way, how do you believe, it's done by this way, why we should change these things in such a short way? Who gives us the right? Well, the right, we want to help the people. And when someone is struggling, we want to be good with this person. So in fact, the probiotic is our wish, good intention to improve the quality of life of the our friends. But again, it's need to be very careful because if we mess at something that we don't understand, maybe we will regret later. So rather do it everything with the appropriate responsibility to be sure that we're not going to create a new Frankenstein. Maybe we can have a break now.
Yes, sir.
Everyone wants a break, right? Okay, please. Actually, I don't know. At 2 o'clock, we need to be here or in the other room.
Yeah.
Okay. Because Santos was mentioning to me something, but I was in one moment confused because. Oh, okay. I know we have a lecture,
Yes.
but again, it was where will be here, there, so here.
Yes.
Confirm it.
Yes.
Okay. So please, I will try to change now to the PDF, and I will send to team of Santos, and then you can distribute to everyone.